Hi, welcome to the Orthobiologics Podcast. I'm your host, Trevor Turner, MD, RMSK, brought to you by Georgia Bone and Joint. We are here on episode 6, which is What is it that's going inside my injection? In this episode in particular, we're going to talk about different applications of orthobiologics and why people get what they get and how they can be confident that physicians who are performing procedures for them are actually giving them doses and medications that are tailored to exactly what the problem is that they are facing. In this episode today, we are honored to be joined by our guest, Dr. David Carley, who is a board-certified PM&R physician practicing at the Stedman Clinic in Vail, Colorado. Dr. Carley is also the founder and CEO of Grayledge Biotech, who is going to talk with us about how he uses a special lab protocol to be able to provide the quality assurance that patients need to get the right injections when they need it. Thank you so much, Dr. Carley. Why don't you go ahead and tell us about who you are and what your mission is?
Sure. Thank you, Trevor. I think that summary pretty much identifies the crux of the problem, doesn't it? And so about 10 to 12 years of my life now have been spent on addressing that very issue that you just summarized so eloquently. And thank you very much for having me on. It's a pleasure to be here and a privilege to work with you in this field as well. To the audience, Trevor is a rising star in this field, and you're going to be hearing a lot more from him in the future. So Grayledge really was designed to address the problem of a complete lack of quality control with regards to point-of-care biologics. And for your audience, what are point-of-care biologics? Well, we're talking about platelet-rich plasma and bone marrow concentrate, as well as some other simple products where we prepare the patient's tissue, in this case, blood or bone marrow sample, process it, and immediately put it back into the patient. For years and years, and even at the start of my career with biologics, physicians use small Class 2A medical devices that would fit at the bedside, where you input a sample, it's processed, and a final product comes out of the device, which is then implanted into the patient. There is no analytics on pretty much every single option that physicians have to use for these biologics in the entire field.
So, Dr. Carley, what do you mean when you say analytics? Can you specify that?
We don't know what we're putting into the patient.
Okay,
right?
that's a good summary. Yep. So keep going.
Yeah. So for years and years, we were trying to validate that PRP and BMC, and even to date, this continues to happen, work. Well, how can you prove that something works if you don't know what you put in the first place? And the analogy that I use is that I think people can relate to is imagine you're studying a blood pressure medication and you have 5 different doses. And your job is to figure out which dose works to correct blood pressure. You don't know what dose you gave, yet your job and task is to figure out what dose is optimal. Right? And that's where we were with this field. If you don't know what you're putting into the patient, you don't know why it worked if it worked, and you don't know why it didn't work if it didn't work. And there's no way without measuring the cells, the number of cells, the concentrations, the ratios of cells, all the different cell populations that are in these complicated biologic products, there's no way to improve on what we did or even explain successes and failures if we didn't know what was in it in the first place. So the premise of Grayledge Technologies was to solve that problem. Let's at least start. In the early days, it was just, let's see what we can possibly measure in these things. And it's evolved, obviously, over the years and become a bit more complex and dialed in. But that was really, I didn't feel it was appropriate nor acceptable for us as physicians to not know what we were putting into the patient, even if it was their own tissue, right? There's not much else in medicine or other areas of medicine where we do that, where we don't know what we're putting into a patient. If you think about a medication or a surgical implant or hardware or anything else we do, we always know a dose. There's always quality that goes into, that we can track, that goes into the product that we implant into the patient. In this case, that wasn't happening. And I didn't feel it was on a level that was consistent with everything else that we do in medicine and our job as practitioners to keep patients safe and give them the best chance for success.
So that's an excellent summary of why your company exists and why you decided to start it. So that, it sounds like you were very committed to raising the bar and also to being able to say, look, there's things that we have evidence for and there's things we have evidence against, and let's give you the best evidence to put your foot forward. It's interesting to hear somebody who is committed to that, because I think there's a lot of people out there who would say, well, look, for me to be able to use PRP or bone marrow concentrate or another biologic, all I really got to do is, talk to a vendor and maybe even take a weekend course, and they'll show up with a machine that's on site, and, various machines are kind of advertised at creating certain amounts of platelet X or, fold over baseline. And we kind of discussed some of those metrics in a prior show when we were defining what is PRP, what is a stem cell, what is bone marrow derived and adipose derived. But the question is, if you're operating that way, or if you're practicing that way, how do you really know what you're getting? And that's something that you set out to answer. So, Dr. Carley, when you spoke about using analytics, how do you, or what kind of process or what kind of machines, how do you start to answer that question when you go to a patient or you look at a sample before a procedure and say, yes, this is in fact the right dose of platelets or cells or whatever that you want to give them?
Well, it was an interesting challenge at first early on that we had to solve. So what's important to measure? There's a lot of stuff in here that at least we could measure, but logistics and cost effectiveness and all those kinds of things were important. But what was most important was to try to put it in a language that doctors understand. So doctors understand red blood count, white count, different types of white blood cells, and platelets, which are primary components of both PRP and bone marrow concentrate. So we felt that if we started by measuring elements that we would measure in the blood normally in the course of medical, that would be a good place to start. And then our task was to prove that those measures were the right measures to be measuring. Does that make sense? So It wasn't, we could measure all kinds of stuff, but it had to be something we could do on the fly. It had to be something we could do immediately, so that when the sample is handed to the doctor, Dr. Turner, here's your sample, here are the analytics. Not, okay, you're going to get your analytics in 3 days or 5 days or 7 days or 8 days, because it doesn't give you the opportunity potentially to alter the course of what you want to do or make an adjustment based on that particular sample. We know that patient to patient to patient, there's enormous variability. So because of that, if you don't account for analytics and processing these things to target a specific platelet count, for example, or a white blood cell count, for example, they're going to be all over the place. And how can we compare one patient to the next if there's no standardization and no quality control going into the biologic? We can't, is the answer. And so we put it in a language, to answer your question, we put it in a language that doctors understand. Red blood cell count, white blood cell count, what we call a differential, or the different types of white blood cells, and platelets. And those were the critical variables. Then we decided to measure them as we got the sample, a blood sample or a bone marrow sample, at the beginning before we processed and after we processed. So we know where we started and we know where we ended up. And why is that important? Well, as we try to understand what works best or what might not work to give patients the best chance for a successful outcome, we felt it was important to know what happens to that sample as it goes through processing. So we ultimately want to be able to draw some blood or take a bone marrow sample, analyze it before we process it, and say, this patient is likely to respond based on our measures, okay, or based on our data. So it creates a predictive model that allows the doctor to counsel the patient that either this is good, it's a go, this is money well spent, it gives you the best chance for success, or wait a minute, something's not right here. We may have to make an adjustment or draw more blood, or maybe we're looking at multiple procedures to give you a good outcome. That's what it's designed to do. It's really a learning model at the end of the day. right? It's designed to learn as we go. And as we generate more and more data, then we can refine the process specific to patient. For example, you might treat a 75-year-old smoker differently than a 23-year-old athlete. Specific to patient and specific to indication. You might treat a bony problem different than a cartilage problem or a ligament problem, an acute problem different from a chronic problem. And the only way we could customize these preparations was to start by understanding, okay, these 50 patients did well. Let's take a look at their analytics and try to determine what trends might be predictors. These patients didn't do well. What was it about their biologic that we might be able to use as a predictor to prevent the non-responders from happening when people are spending their hard-earned money in a cash-based model for these procedures?
Some things that people have talked about include, age. You mentioned as one big variable that's out there. And, you mentioned a couple of other things like maybe smoking or, that people, when they come and ask, well, how do I know that my cells are as strong as they could be, or how do I prepare for a procedure? Um, when you start talking about variability and the importance of measurement, I mean, can you give us an example of some surprises or maybe some outliers and somebody who's, older who tends to actually have, a great cell draw or something else that surprised you when you were doing your processing and analytics?
Yeah, we just had this recently. So the common notion is that as we age, we don't have, number one, we don't have any stem cells, and the stem cells we have are no good. Well, as some of my PhD colleagues in the field would say, if you didn't have any stem cells, you'd be dead, right? Because stem cells are what keep us alive and maintain our bodies, right? So that is a myth. Now, stem cell density or concentrations of stem cells in our bodies do decline with time. But what we've found, and the majority of patients we treat in like an arthritic model, for example, aren't in their 20s. They're in their 60s or 70s or 80s. And we track all of those numbers, obviously. So we're seeing that we can concentrate, through processing those cell counts up to a nice, high, robust range. And this notion that older people don't have good stem cells really is not true. If you look at Philippe Bernigo's work out of the University of Paris in France, Philippe did a lot of work in people over 80 years of age in arthritic models using bone marrow concentrate, and was able to show that they had favorable responses just like younger patients did. So it was groundbreaking work, and that has kind of mimicked what my practice has seen, and I think yours possibly and other colleagues in the field are seeing and reporting, that just because you're 70 or 75 or 80, if you're healthy and in good health, we still should be able to create a product that gives you a reasonable chance for a successful outcome. We just had a patient recently, you and I talked about, where we had a extremely high cell count. Now, she was a skier, right? She's still very active and skis, 50, 60 days a year and exercises and she won the genetic lottery in some respects, but she's in her 80s, and we had a very, very high white blood cell count that I think I mentioned to you and to some of my other colleagues. So yeah, it exists. The issue is, what happens if we draw a bone marrow and that count is low? Then what, right? Does that matter? Should we still use the sample? Should we add PRP to it? So it creates a decision-making, model ultimately, where if this happens, then do that, okay? If the numbers are great, everybody's happy and we're moving forward. But if you're not measuring, you wouldn't ever account for those potential issues, which we do run into. Or what if a sample clots? If you're using a device and not measuring, that platelet count in PRP goes almost to zero. Yeah. Now we're injecting not PRP, but something entirely different. So it's quality control to make this more drug-like, okay? Dose response, concentration of cells, and response. And then what we can do over time is, as that model continues to be refined, we park it in databases along with patient-reported outcomes, where the patient is telling us how they're doing. And then we can start to link and merge the two data sets together. And that really is what Gray Ledge was ultimately designed to do.
Great. So it obviously sounds like a huge part of your mission is being able to sort of optimize or make the practice of biologic or orthobiologic medicine, a new standard that has a lot of strong evidence behind it that's linked to good outcomes. Some of the other questions that I'll get from my patients are, is this covered by my insurance? And then, the answer is no. But, typically it's then, well, when is it going to be covered? When can I expect to get it to be covered?
Yeah.
And a lot of what I tell them is, that's the $1 million question. But I think if we're ever going to get to a place that we're going to hope to have this covered, then the way to do it is to collect outcomes data and do rigorous scientific analysis so that we can actually say, look, for everybody out there who says there's not evidence, we have emerging, decades and decades of evidence that are getting stronger, as we collect this and as we correlate it with patient outcomes. Can I ask you, so since you've been doing this, at least in the US, a long time, I would say, you mentioned kind of one of the, maybe the progenitors or the original, the godfathers of biologic medicine and Philippe Pernigo. But since you have been collecting data, and looking at analytes for 10 years, in what ways, or could you describe a little bit how the pattern of the way that you practice biologics and orthopedics has changed as a result?
Yeah, I wish it was a bit more specific than it is, and that's getting better and better. But we have identified that we can hit targets from a processing end. So we can say, so in PRP medicine, for example, some people believe you should have white cells, some people believe you shouldn't. Well, if you don't measure, how do you know if you do or you don't?
Sure.
So an important piece, even before we were able to observe, is can we do it? Can we make a PRP with a high platelet count that doesn't have white blood cells and has a low red blood cell? So we can change white blood cells, we can change red blood cells, and we can change platelets. 3 variables that give us the potential, at least in theory, to customize. So the first thing we learned was we can target a custom prep. Now, BMC is a little more complex because it's still kind of more is better type of approach, but ultimately we're going to work on refining that one as well. For example, I'll give you a blood cells in BMC. We seem to feel collectively as a group that we don't need them in PRP, yet our red blood cell counts in bone marrow are very high. So how do you create a bone marrow preparation potentially that has a low red blood cell count? That's something we're working on, okay? Another thing we're working on, and this is very new, is how do we optimize the cells? So we talked earlier about, stem cell densities decreasing with age. Well,
Right.
what if we could make your cells quote unquote young again, or more robust, or more powerful from a therapeutic standpoint? Something we can do systemically. It could be supplementation. It could be improving the medical condition or the system, the environment with which those cells live. That's a separate project we're working on, is how do we prime your cells to make them work better for you? Okay. So all of these things were ideas and concepts, and we didn't know if it was even possible to get a model working that could answer the questions. Is it possible, and does it matter? So we now know it is possible. We now know the measures that we are collecting are validated. So there's a lot of testing that goes into our instruments and to make sure that what we're measuring is accurate. Because if you measure something on an instrument that's not accurate, it really doesn't mean help or meaning. So there's a lot of work that went into, this was the difficult work that went into creating the process that could answer the burning questions in the field. So we went from, let's start by, can we measure something? What's important to measure? Is that measure accurate? Can we alter those measurements or target a preparation? And now you're starting to see the model form and morph that we will be able to target a problem specifically, target a patient population specifically, as we continue to learn about what works best and what doesn't work. We can learn as much from our failures as we can learn from our successes. It's great when you do a PRP and everybody's happy and it works. No one likes spending money on a biologic that doesn't work. There's an opportunity to learn with every single one of these we do, positive or negative. And I think, as physicians, we tend to not want to spend time with our failures. right? We want to spend
Sure.
time with our successes, but we have to spend time with the failures because the opportunity to learn there may be even greater than with our successes. Okay.
Absolutely. I think most of the talented people that I've met who've achieved great things have often said they learn more from their failures than they do from their successes. One of the things that you mentioned was process. You use that word a couple times, and I think most people just think, okay, well, once I have the blood or the bone marrow cells, I put them in a disc or a container, and I stick them in a machine, and it spins around, and then my output comes out. Your process has really become much more labor intensive than that.
Yeah.
So requiring the use of a technician, requiring some really special things in order to ensure that you have a sterile protocol, even when it comes to transportation of samples. Because I know in your practice, similar to ours, we use a lot of biologics in the operating room as well as just without a surgery percutaneously or less invasively. So can you talk a little bit more about what it requires to ensure that everything is done sterilely and make sure that the processing is done to the standard or the specification that you're looking for?
Yeah. And it was quite a process, as you know. So our platform actually uses a processing laboratory as opposed to a medical device. Well, why do that? Because it's immensely more complex and difficult to operate as a business, right? And as a service. Well, the reason was twofold. One, we knew that the regulatory environment in this field was dynamic. It was changing. It had potential to change. So we create a medical device and the regulations change. Now you've got to totally change and reverse engineer that device to meet the regulations. In a laboratory setting, we can manipulate or change things to mature with the field that is dynamic as it changes. This is better. Okay, we can change an operating procedure, or we can change a quality management program or protocol. So it was much more malleable to grow with a dynamic field. So we can't do that with a device. Lab makes more sense, even though it's way, way more difficult. The other thing with the lab was and we're still learning this, what is critical to measure, right? Are there other things we should be measuring? So once you build a device, you're married to that device and its capabilities. And the devices can't really change a lot, because if you change a device, then you got to go back and get approval to market the device. In a lab, we can always adapt. So I said earlier that this was a learning environment meant to help us learn and grow and optimize as clinicians and as a field. The only way we felt we could do that successfully was to have a much tighter control and the ability to change as the field changes or as what we learn changes. So it required, technicians processing manually, hemoanalytics with analyzers that measure our samples, registering with the FDA, going through audits with CBER and the FDA twice. Because the other thing was, in a challenging regulatory environment, we wanted to be transparent from day one, so that if we were doing something wrong, we found out early and had the opportunity to change it, rather than bring this company along. We've been doing it wrong the whole time. Now the FDA comes in, barks at us, and we're scrambling to try to fix that problem. And that's happened to many companies. Okay. From day one, we registered with the FDA. We said, here's what we did. We set up our programs to surpass what we felt were the FDA's expectations. And it did. It performed. And that was the best thing I ever did, was to be transparent and upfront with them from day one. And that's hard, right? This was the harder road for sure. And it slowed us down as a company to create a product that was competitive with the devices, but also was efficient enough to park in a practice that wouldn't screw up the workflow of busy physicians. So they were enormous challenges and enormous hurdles, and it took us 10 years to refine it and hone it. And we did all that through my practice and my colleagues' practice here in Vail. And it only in the past maybe year and a half got to the point where we felt we could scale it and put it in other locations, one being at Georgia Bone and Joint. And the reason for that is we said it's a learning model, but that learning model only works when we're collecting large amounts of data. Well, now we had a system that could compare apples to apples to apples, no matter where it was, because the protocols were the same and the analytics were the same, as opposed to, well, this group uses this device and this group uses a different device and none of them are measuring anything. How do we compare one center to the next? So we had to standardize at least the process of how we make these preparations so that we could compare in multi-center fashion. Now, what we can do, at least in theory, is to collect patient samples of 1,000 or 2,000 or 5,000, as opposed to 20 or 30 or 50, which have governed our field in terms of the research to date, very small patient samples. Well, as you know, the statistical relevance of those studies are not where we need them to be. We need success in 5,000 patients to really observe trends that are going to allow us to tease down and refine these preparations and make them better over time.
Yeah, absolutely. And that's why, I mean, I would thank certainly my partners here at Georgia Bone and Joint for being committed to that notion that the only way that you truly advance the science forward and you get those statistical numbers that enable you to have enough power to be convicted when a patient comes in your room and looks at you and you're looking at your MRI or an ultrasound or x-ray with them and say, look, based on our evidence, this is what you can expect, and to understand which things are likely to help and which things are not, and to be able to approach it in a way that is ethical. So when you started forming a team together, I mean, it's interesting because you say, well, we have techs and, well, we had a PhD and, we invited in the FDA, which obviously we commend you for, because they're, people joke about certain of our wireless cell phone companies saying, well, this company does it first and this company does it right. But can you talk about kind of how you assembled your core team, or who were really instrumental in helping you achieve this mission and this goal of yours?
Yeah, well, they were humble beginnings. I remember in the very early days looking through a microscope, counting cells, which is mind-numbing work. But that's where we started, and it obviously has advanced as we were exposed to more technology as we grew and matured ourselves. But there's an element of this that required regulatory expertise. So we went after some of this. We outsourced, but some is insourced as a company. Knew needed someone who had a good grasp on the FDA and what they wanted from us, what they expected from us, because that's a really challenging area in this field. We wanted to meet their expectations, so we had to get expertise in that. We continue to grow our presence from a research standpoint, PhD level. So we have a full-time PhD on, and I'm courting a second to come on to our company as well. And so we figured out, okay, well, we can measure. Well, what about all this data? Someone needs to take that data and start to dig into it. That requires statisticians and researchers and quite a bit of brain power and manpower to make that into something that is useful that we can learn from. So again, another challenge. Well, let's figure out how to measure. Okay, we did that. Let's figure out how to collate that data. Okay, we did that. Let's park it in multiple locations. We did that. Well, now we have all this data. What are we going to do with it? So we needed to go after resources to handle that. On the technician end, we are only as good as the product that our technician can produce. So it became a critical position, especially in remote sites, when we're managing things from Colorado, but we have a technician in Georgia, and we have to make sure you process it on the same level that we do. And same for, another state and another state and another state. So there was a managerial component to look at quality and auditing ourselves in lab to lab to lab. So there was another piece that we had to bring into this. And it just one built on the next, built on the next, built on the next as we identified the challenges, matured as a company, and then said, well, okay, where is the most value that we can give to clinicians from this company? At the end of the day, it's in the two things we talked about: customization based on patient and customization based on indication. So all those moving parts are geared towards those ends. And are we there? We're getting there. We're getting better. We're growing. And the more people that use this, obviously, the more data we can generate. We're still working on how how to collate the analytics from the sample with outcomes and marry those two things, those two data sets together. But it's accelerating more now. It was kind of a very slow progression and very slow process. And what's happening now is we're starting to see more of a higher slope, if you will, on the growth of what we're trying to do, because we need users to make this thing valuable to the field and help the field, which is what it was designed to do originally.
Well, obviously we're biased, but we want to thank you for taking the initiative to not only start this journey when you did over 10 years ago, but to be committed to such a high standard, because my partners and I at Georgia Bone and Joint, we certainly feel not only do we have the most, advanced laboratory, certainly for the use of orthobiologics here in the Atlanta area, but also the state, and that we're partnered with such a strong company like Gray Ledge Biotech that really has been doing it right from the beginning. How many different centers or sites do you have now, Dr. Carley?
I believe we're in 6 states, and we're courting a 7th. Again, we're growing, but it wasn't just about how many labs we had. It was more the quality of the affiliate that we were working with. As you know, there's a stem cell center on every corner, in metropolitan areas nowadays. We knew we had a powerful vehicle here, but we also knew we needed to get it in the hands of responsible people that we knew we could work with, that had their heads in the right place in terms of where are we going with all of this. Everyone understands the great potential. The public and consumer demand is very high. They want us to come up with solutions. So it wasn't just, and this was another thing that was challenging from a company standpoint, because it would have been easy just to put these labs anywhere with anyone.
Sure.
We didn't take that approach. We wanted people that I trusted were going to use this tool in a responsible way to help us get better, not just to make money or to create a competitive service in a market. So that was important to me, and Georgia Bone and Joint and you in particular were very attractive. I mentioned earlier that we need more Trevor Turners in this field, and Georgia Bone and Joint, to your partners as well, because you've recognized the problem. Okay, we've got a great tool here, but there are some issues with it. You've now committed yourselves to be part of the solution, as opposed to taking the easier road, which was, well, let's do it as well as everybody else, and compete for patients in our marketplace and be able to counsel them, well, we're doing it as good as anybody else. No, you've committed to, we want to do it better. We want to do it best. And we want to create a scientific validation so that when we're talking to our patients, we can say, we are doing research. We're doing this at the highest possible level. And no matter what, we always are committed to learning from you to make it better for you and your family and your friends and other patients who we're treating. That's our commitment. And it's harder to do that with a lab than it is a device. So I always have a lot of respect for anyone who is willing to take this on, because it's more cumbersome and more challenging. It's getting better. It's getting more efficient. But it takes a commitment from the practitioners to say, the status quo isn't good enough. We are cutting edge physicians. Our job to be on the cutting edge is to make what we do better. And this vehicle is designed to help you do that.
Well, Dr. Carley, that's going to wrap us up for about the time we have today, but we want to thank you for your leadership in this field, and thank you for your commitment to helping practices around the country raise the bar. If you are interested in partnering with Dr. Carley and Gray Ledge Biotech, you can definitely check them out on the internet at grayledgebiotech.com. This is the Orthobiologics podcast. That was episode 6. Thank you so much for listening. If you have other questions or you want to find a facility that holds itself to this standard, please look us up on georgiaboneandjoint.org or book your appointment conveniently online. Thank you so much. I'm your host again, Trevor Turner, MD, RMSK. We'll see you next time.